Our hypothesis was centered around the idea about stomatal density with sunlight. We decided to test the stomatal density of plants when sunlight is affected. Stomata directly affects the amount of photosynthesis that each plant goes through. Stomata is the guard between the exchange of carbon dioxide and water between the cell. Our hypothesis is that we decided that plants that receive more sunlight will have a higher stomatal density compared to plants in the shade. We believe more stomata will be open because of the increase in exposure to sunlight compared to the plants in the shade for a prolonged amount of time. Our idea behind providing the hypothesis would be to use two specimens of the same species of plants. If we use the same species of a plant, it eliminates other possible variables that could make the data incorrect. The Zebrina plant would be the plant used. We will use light microscopes to get a clear view of the stomata. By using a light microscope, we can get a view of the stomata. We will begin on a low magnification and then increasing the magnification and adjusting the light to get a better view. From there, we can collect our data and make an estimate of the stomata density in each plant. Stomata density is found by using the number of stomata per millimeter square. We would keep time constant at 2 hours. We would make sunlight the independent variable and stomata the dependent variable. Therefore saying, sunlight would be the x variable and stomata would be the y variable. Sunlight will be the factor that changes, and it will affect the amount of stomata that is open or closed after 2 hours.